to SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode 27. My name is Scott Okuno. I'm medical oncologist at Mayo Clinic and chief medical officer at SARC. At SARC Talk, we interview interesting people who are connected to sarcoma, where we can all share and spread the knowledge about sarcoma. Today, our guest is Dr. Danh Truong. Dr. Truong is Instructor, Sarcoma Medical Oncology at the University of Texas MD Anderson Cancer Center. Dr. Truong received his BS in biology at the University of Texas in Arlington. He also had his master's in bioengineering at University of Texas Arlington, and he received his PhD in biomedical engineering at Arizona State University. He did his postgraduate fellowship at Sarcoma Medical Oncology at MD Anderson, and he's remained at MD Anderson, is now instructor in Sarcoma Medical Oncology at the MD Anderson Cancer Center. Dr. Truong is very active in sarcoma research at MD Anderson, as well as within SARC, and we'll learn more about that today. Please welcome Dr. Truong. Dr. Truong, please share with our audience how you decided and dedicated to pursue a career as a scientist researcher, and then in addition, how you decided to spend your career in sarcoma research.
Yes, of course. Thank you, Dr. Kuno, for the kind introduction. I think, I have I don't know if it's a typical story, but growing up, every day I'd come home from school and as a kid, watched a TV show called Bill Nye the Science Guy. It's basically a science educator on TV on PBS Kids that talks about the history and science behind everyday aspects. And thinking back, I don't know how old I was, probably 8 or 9. Some experiments that occurred on the TV show was sugar crystallization, and I essentially try to replicate that, where you boil water, add some sugar until it's super saturated, let it cool down, add a string, and watch the sugar crystals grow on the string over time. And I still remember. I don't know why, but that was my first experiment as a child. And so, long story short, I studied biology and bioengineering, as you mentioned. And during my PhD, I focused on tumor on a chip models, where you're trying to basically understand tumor interactions at a cellular level for breast cancer. And this, of course, is a long way from the sugar crystallization experiments I did as a child. And then I took an R boot camp at some point during my PhD studies to really start diving into computational biology, because that's where I wanted to go. I wanted to do more sequencing, transcriptomics, genomics, and this led to me applying for a postdoctoral fellowship. Of course, I applied for MD Anderson. This is back in August, I believe, 2018. And unfortunately, I didn't hear back until I think November, and I was graduating in December, so that was like a month in between. And I already had an offer and a couple other interviews at the time, but I said let's go for it. So I went for the interview early December, and I met with Joseph Ludwig, my I guess at that time potential mentor, as well as the Sarcoma Medical Oncology family, and who I thought they were very warm and kind, and I really enjoyed that. And I shared some of my vision on how I wanted to combine like tumor on a chip, as well as computational biology, and for sarcoma to really do a deep dive into biology and find various targets that we can try to treat. And so eventually, of course, I accepted the offer. They gave me the offer, I accepted the offer, and I started in January. So it was a very quick kind of turnaround. And at that time, I had no idea that, what I was getting myself into, in terms of like going from a more common cancer type that everyone knows about to a very rare cancer. And everyone kept telling me, there's low hanging fruit everywhere. And, and still to this day, I hear that phrase, but things are never that easy. There's a lot of stuff to do, and it's still challenging. But I like it. I love it. I feel like I can make a really big impact in this field.
So Don, you said something that I'm just going to go back and push a little bit. There must have been a decision with the success of your sugar crystal on a string that said, I want to be a scientist that does research versus I want to be a clinician and see patients. Now, I'm not saying that the fork in the road is that black and white as we think about it, but you knew earlier on that you wanted to be a scientist and work on understanding and doing that work. Um, Other than that science fair, not science, your home experiment, there must have been something else that kind of said, "This is, I enjoy this."
No, I mean, of course, I could go through more stories. I actually did participate in science fairs, and of course, but I think the thing that I've done a lot of self reflection, but the thing that I think that my trait that kind of hones me on into the scientist is curiosity. I'm a curious person. I like to push buttons, turn knobs, figure out what's going on. What happens if I do this? What happens if I change that? And then it's kind of embedded in me, and I feel like that's a lot of the spirit of being a scientist in a way, where you're curious about nature. You want to know why these certain things, these biological phenomena are occurring, and really unravel the complexity there. And I think that's the trait that I have that that kind of like basically focused me on this path towards becoming a scientist.
Yeah. No, that does make sense. The curiosity and testing to see if that works or not, that makes great sense. You started doing breast cancer research, and then when you were finishing up, you said, let's just go to MD Anderson, and you met a group of people, Dr. Ludwig and the sarcoma team. That was just a stroke of luck, or how did that happen? Because what you shared is what we think all sarcoma investigators or clinicians are. They're just warm and welcoming and collaborative by nature. And that's how, whether it's MD Anderson or any other place, I think sarcoma just lends it to itself. So how did you end up at the sarcoma part of MD Anderson? Because you could have gone to MD Anderson's huge. How did you end up there?
Yeah, it's a good question. One is I'm from the Texas area. I grew up in Arlington, Texas, so that kind of goes back to some of the schooling I had at University of Texas at Arlington. The second aspect is some of Dr. Ludwig's work had to do with growing Ewing sarcoma cells in 3D culture. And I had read a review article at the time. It was actually like a review article about 3D culture for sarcomas. And just by happening to read that article, and I saw there was a PI at MD Anderson working in that space, and that was like sort of a bioengineering space, right? That's where I was coming from. I thought it'd be an exciting sort of cross section with the work that I've been doing, and how I can bring some of that to MD Anderson. I wasn't sure at the time. Sarcoma wasn't on top of my mind at the moment, but at that time it was mostly about the engineering, the 3D culture.
Yeah, that's exciting because a lot of the people we've interviewed in SARC Talk kind of done it the same way. They stumbled upon it. However, there are some that they knew when they were in grade school they want to be a sarcoma person, which is okay. Now to MD Anderson, you have a lot of opportunities for collaboration within the sarcoma group. We had Dr. Nassif Haddad in a previous SARC talk, and she talked about the leiomyosarcoma working group that you are also engaged with. I do know that you and Dr. Ludwig work very well together as well, and have mutual interest in developing certain strategies within sarcoma. And maybe you can share with our audiences, once you're in the sarcoma group, how do you maintain collaboration but also focus? Because otherwise you could be going all over the place. So how have you been able to um, work with the sarcoma groups and multiple different investigators?
Yeah, I think that's where you gotta have sort of a niche and an expertise that you sort of bring to the table. And then, yeah, of course, I've been working with the various sarcoma working groups. I think I came to MD Anderson in 2019, and that's when Joe Ludwig invited me to join the liposarcoma working group that was led by Nita Samaya and Barry Feig at the time. And I guess part of that also transitioned to what you were talking about, the leiomyosarcoma working group that's led by Elise, Emily, and since then, I've actually joined several other working groups as well, such as this one called, I wrote it down here, CURE OS, which is a collaborative understanding of the range and evolution of osteosarcoma single cell. So it's a long word, so we just go CURE OS. And it's basically work in osteosarcoma. It's led by MIB through Christina Iptoma. And I really enjoy my time working in these various working groups. And I think, to your point earlier, like how can you be part of all these working groups? I think you have to really know what your expertise in, what your niche is, and how you can, what kind of basically contributions that you can provide, right? So obviously, I'm not going to be the one that's doing all the experiments, all the research, but I attend obviously the seminars, the webinars, and provide insights on single cell biology, spatial biology, some of the things that I know that I'm well versed in and that I can contribute to these topics. And speaking of like how I came about that, I think of course that most of that is really due to some of the mentorship I've had with Joe Ludwig. I mean, he really uh, helped me build my career, and he put a lot of trust in me to develop and find my niche. I think when I came here early in 2019, one of the things that we were trying to study was differentiation errors in sarcomas using mesenchymal stem cells. And we were thinking, hey, we're going to try to find the expression of 200s or 300s of proteins. And we're going to do like 1,000 Western blots it was very tedious work, essentially. And at that time, I read an article about single cell RNA sequencing where we could take each cell one at a time and look at their individual profiles. And why that is important is because when we're differentiating these cells from MSCs to whatever cell, osteoblasts or adipocytes, for instance, some cells differentiate a little bit slower, some cells differentiate a little bit faster. And when you do a Western blot or a traditional RNA-seq, you end up just averaging their profile into a single expression value. And you don't really get that granularity of the slow cells, the fast cells, the cells that are in the middle. Actually, you only get the cells in the middle, right? Not the cells at the other time point. So I brought that up to him, and I guess I was able to convince him because to this day I've been doing a lot of single cell analyses for so many different projects, and it's been part of the mainstay of what I do right now.
Yeah. Well, as you said, your core, everything you have in your knowledge set could be applied to a lot of different sarcomas, and that's why you work in multiple sarcoma working groups and you learn and leverage. Yeah, there are different biologies, but you did bring up something interesting to all of us is that single cell analysis is a key component because it's not the average of every single cell, it's there. And those that are listening, you might have heard MSC, and that's a mesenchymal stem cell. Is that correct? So just in case people are hearing, what was it MSC that Dr. Trong said? But that is correct. So we're excited. Now there's some similarities with you and Joe's path, but we'll come to that in sarcoma here. Um, we really enjoyed your presentation at the last SARC meeting in ASCO in 2024 in Chicago, where you were talking about your studies that you were doing, that you and Joe were doing there. Um, you are a member of SARC's Discovery Translational Committee, and maybe you can share with our audience how you got involved with SARC and how it's been working within the SARC Discovery Translation Committee?
Yeah, I remember. We had the COVID pandemic, and then it was right after. I think everyone went to CTOS in Tokyo. I believe that was the year, and then I didn't. Unfortunately, I didn't get to go that year. And then ASCO, I'm sorry, CTOS came back in 2022 in Vancouver, and there I attended my first SARC meeting, and it was an amazing experience to be there to see all the folks that are involved, from researchers, clinicians, patient advocates, and there was a part of that session, I believe, there was a mention about the Career Development Award from Jonathan Fletcher, basically developing the careers of young investigators involved in sarcoma research. And he mentioned that PhDs, if or would could also apply as well. You didn't have to have an MD; you can have MD, PhD, or a PhD. And Joe, who was sitting next to me at the time, looked at me and said, "Hey, Don, you should probably apply for that." And of course, I went to the informational webinar that came afterwards. I wrote the LOI, submitted that, and the rest is, as you know, I was awarded the Career Development Award in the very end, and that really started my journey of being really involved with SARC, joining the Discovery Translational Committee, and it also helped me really understand where I kind of was in my in the broad, big picture sort of thing, where things that I do, my research can, I think, have an impact on patients, have an impact on how clinicians think, and have an impact on perhaps trainees down the road, when they want to study sarcoma. Um, you know, before that, I think I didn't realize how far some of the things I do, the research that I'm involved in, uh, I wasn't sure of the impact at that until then.
Yeah. So for those that are listening to or watching, Joe Ludwig also had a Career Development Award as well. So we have a duo now coming from that lab there. And I think what Dr. Truong and even Joe, if you were here today, it really catalyzed or catapulted them into the sarcoma field and encouraged young investigators to stay in the field of sarcoma, which almost all of them have, which is great. And Dr. Truong, what you said is interesting and important is that it put a clinical face on what you do. You're still doing the nuts and bolts research, but there's a clinical aspect of, hey, this I'm doing here will impact patients down the road or pretty much immediate. And we, SARC is very proud of the SARC CDA. We're working hard to get that back. So if anybody's listening is interested in learning more about supporting the SARC CDA, please let us know and we'll be happy to connect. The CDA is complete now, right? It's for 2 years, right?
Yeah, well,
Or 2 or 3 years.
I think it was 1 year.
Yep. But you're out now a couple years, so it's all done.
No, we do have a little bit left, so we did request a non-cost extension. So we're
Okay.
trying to complete that.
The little bit of detail there, which is important because, part of this is to say is this is a career starter. Now we got to continue to work and make Dr. Trung more and more because he has now the skill sets. He's passionate, ready to go. And that's why he's engaged in the sarcoma SARC Discovery Translational Committee. And then that committee, along with a lot of other people, have put together a yearly meeting called SAS, Strategic Advance in Sarcoma Science. And they've met now twice. And maybe you can share a little bit about what SAS is for people that are listening, and then we'll ask you what SASE means. But we'll start with SAS first.
Sure, yeah. So SAS is, I think, a wonderful research meeting for sarcoma investigators. We really, in that space, we don't have a similar meeting type. I mean, I think it came when I spoke with Jonathan Fletcher, who was the one who really developed SAS. He said it's similar. It came from, he was inspired by the Fortress group meeting, which is a European counterpart, where they're also looking at the basic science and translational science of sarcoma. And I believe they try not to talk about any clinical science at all. It's all mostly basic and translational. And he was really inspired by that and wanted to have something similar for folks in the United States. And I guess it also comes along with AACR. AACR also had a sarcoma conference, but I'm not sure if it's coming back or not. But it's every other year or every 2 years. But he wanted something that was annual, and he wanted something that's very small and intimate. And when I say small, probably around 80 to 120 uh, investigators, ranging from young investigators all the way to senior investigators. You have a broad range of expertise and really be able to bring all these folks together. And currently it's happening in DC, but bring them all together on the campus and share some of the latest unpublished science so we can discuss, help each other, and really generate collaborations and move the field forward.
You're right. And the unique thing is Dr. Wiedemann and Jonathan and the NCI have really put together two great meetings, but it's really basic science and translational. And by design, you want it to be interactive. You want the people that are there that are pure scientists working on sarcoma science and biology. And unfortunately, in order to be small and collaborative, you have to limit the numbers. And I think, so we just need to understand that. And I know that the Discovery Translation Committee does vet the agendas, the people, and whatnot. And hopefully, that if we had our druthers, everybody would go, but then that would diffuse the meeting. Now, there was a component that arose that, yes, as Dr. Trong said, there are mixtures of senior investigators, middle year career investigators, and young investigators. There was this feeling that there was a younger investigator group that needed a little bit more togetherness time. And then you came up with the word, or somebody came up with the word sassy. So maybe you can tell the group what sassy is.
Right. And so this came out of a breakout room we had in the original SAS meeting in 2023. It was basically developing resources for sarcoma investigators. It may have been focused on young, but it was developing resources. And we had three different broad aims. One of them was a young investigator forum. And we debated, quite a bit whether it should be through Zoom, basically online, or hybrid, or should we have a local meeting. I think at the very end, we decided to have that sort of like a in-person meeting and tag it with the SAS. So usually SASC actually stands for SAS Young Investigators. So that's why it's SASC. The Y comes from Young Investigators. And I was actually in the room when we came up with the word SASC. I didn't come up with that. That was actually a Corey Weissstuck, I believe, who's at MSK. But now it's stuck, and I guess everyone calls it SASSy now. And so yes, so we wanted to have something that was logistically manageable. So that's why we tagged it with the main SAS meeting. So usually SASC, I'm sorry, will happen about either most likely the day before SAS. So most of the young investigators will come 1 or 2 days early, and they'll have a separate meeting focused on career development, as well as some research, and where folks can both meet with one another. And we had our first SASE in 2024, so the following year. And I think one of my takeaways, not just beyond the career development portion, but there was a social hour that day, uh, and where all the young investigators, I think there were probably 20 or 30 of us, and most of the time, when young investigators go to meetings, it's a broad range of folks, young investigators, middle career, and then senior, as you say. But we only really know the middle and senior folks. Maybe we've seen their talks, we've seen their papers, or we collaborate them, but the young investigators don't know each other. We've never met, obviously, right? And so this meeting, SASC, gave us a social hour where young investigators got to mingle, talk about their science, develop relationships. And I think it's important because now, if you think about it, these sarcoma young investigators will hopefully stay in sarcoma for the next 20 and 30 years. But now we know each other at very early, onset. So now, we can start thinking about collaborations, things that we want to do together for a really long time. I think that was one major takeaway that I got from the SASE group.
It works as designed. If you get 20, 30 people that are interested and know each other, you're the base that we're going to expect a lot from now. And you guys will know each other. And this is a great thing. And it's a constant renewal. There'll be a time when you no longer sassy. We have to talk about a mid-career name, okay, or senior career move. But the reality, it's exciting, and this is exactly what we're trying to do, is to make sure that SARC is supporting the young investigators' careers in sarcoma research, and this is one component. As I said last year at our SARC meeting in Chicago, we heard your great talk on the androgen receptor, and that's the clinical facing component. There's a lot of basic science in there as well. And maybe you can share kind of why should we talk about androgen receptors in sarcomas, how that came about, you and Joe, and then more importantly is what type of sarcoma does this apply to, and maybe you can share with our group on that.
Of course, yeah. So we were really studying this disease called a desmoplastic small round cell tumor. It's a soft tissue malignancy that generally arises within the abdomen, within the peritoneum. And what it looks like is really these small round cells that are surrounded by heavy desmoplasia, and occurs in predominantly male. Usually, 9 out of 10 are going to be male, and unfortunately, the overall survival is 5 year survival is between 15 to 20%. And one more thing to mention, that it's an ultra rare cancer. Usually you'll have between 30 to 50 cases perhaps per year in the United States. And so not much is really known about this disease, but some of the work by Andrea Hayes, who's a pediatric surgeon, she was a pioneer in really treating DSRCT, and then she noticed especially that besides that most of her patients were male, that DSRC seems to occur near the reproductive organs of a male, and then she got her wondering whether DST was um, hormonally driven by androgens. So she looked into AR, as well as our group looking into AR, and of course, DST ended up being quite high in androgen receptor expression. And our group with Joe Ludwig really investigated the function, whether we could stimulate DSRCT. At the time, we really didn't have much to go with because we only had one cell line, I believe, and one mouse model. We showed that some androgens were able to stimulate the growth of these cells in the lab. And the critical point of this is that AR is really important for another disease called prostate cancer. And so with prostate cancer, there's several different types of drugs that can be used to treat, but those that do are targeting AR, such as anti-androgen and anti-AR, such as enzalutamide. We may be able to use those in DSRCT. Some of our work has also shown that AR is also heterogeneous, meaning that that it may be present in some cases and may be absent in the other. So we're still trying to understand, okay, if it's important in some and then not the other, how can we really treat those other tumors that are not presenting AR? But with those that do present with AR, we're really trying to study other cancers, such as prostate cancer. Some of the literature research I've been doing looked into work in melanoma, breast cancers, which also seems to be relevant for AR as well. We're trying to look into understand how we can leverage some of those science from other more common cancers to treat DSRCT.
So an ultra rare tumor that might have therapeutic implications, not only for desmoplastic small round cell tumor, but other sarcomas as well, or other tumors as well. And that's the nice pivot that we're doing. And I know there's probably some thinking going on and designing more robust clinical trials, but there's still a lot more that we need to know because desmoplastic small round cell tumor, it's more than just the androgen receptor, just one component. And
Of
we
course.
look forward to hearing more about the story on it because it's an ultra rare tumor, and that's the good part. The other aspect of being a young investigator and going to SASE and being able to present at a big meeting for us to kind of get your name out there, the people know about you. The issue is, what else do young investigators need or want that's lacking to build a career in sarcoma? During your social hour, during your SASE meeting, you guys must have said, if we had X, I would stay more in sarcoma or more stay in research. But since I'm not that researcher and I'm probably too old to be even called sassy, where do you see, if we had time and effort, where should we be spending our efforts to support sassy like investigators to remain in sarcoma research?
Yeah, I think that's a really important question. It's also something that I'm facing myself. One of the important things is, especially being PhD only, my sole focus really is research. And so one of the resources, of course, is really funding. How can we secure funding for cancer research, for sarcoma research, and for young investigators? And oftentimes, I've faced this, and I'm sure many other folks have faced this. We submit research proposals to some of the larger institutions, such as the NIH, for instance, and we hear these comments such as like, the impact isn't that wide. These cancers are too rare, and it disappoints me. But I still push on. I mean, and then I work with foundations such as Sarcoma Foundation of America, as well as SARC, where there is a sort of initiative to fund a lot of these sarcoma science. So that's some things that I myself have been trying to reach out for. Um, Another resource aside from the funding is I actually, perhaps myself, for young investigators and those trying to get into sarcoma science or wanting to stay. I really enjoy the podcast. And recently, as I talked to you, I think last year at CTOS about SARC Talk, and being a big fan. And actually, when you invited me to join SARC Talk, I had not listened for a while, and I decided to go back. And the first episode I listened to was the one with the research advocacy group with Val and Caroline, and
Oh yeah,
Val,
yeah.
who was whose son was affected by DSOCT, and you did mention that you attended a recent meeting about the researcher who talked about DSOCT at ASCO, and I was like, wow, that's kind of serendipitous because you literally just invited me the week before. And when you invited me, it was back in like January or February, so that would have been like 6, 7 months later. But yes, these podcasts I find really inspiring. I also listen to Sarcoma Sisters, Sarcoma Insight, and it gives me a different point of view from just the research side, but also the patient advocacy side, the clinical side as well. So those are other important resources I think all sarcoma young investigators should be, looking into as well.
Yes. Well, first of all, thanks for coming on, first of all, because it takes some time to get people involved. Part of our job is to bring awareness to the importance of sarcoma. And that's exactly what we're trying to be, advocates for rare tumors. Because as you know, sarcomas account for 1%. And if you're doing desmoplastic small round cell tumor, which you have less than 100 cases a year, who's going to think that's important? Well, you saw Val, it is important if it's you or your loved one, and we want to be able to do that. So yes, let's get the word out, share the importance of sarcoma, ultra rare tumors, and that's a common voice. So please share. Your component of funding is even more relevant today as groups look to evaluate where we're spending money, how we spend the money, and how efficient we use our money. We have to still articulate we are using money wisely and it's money well spent. You shared that what you're learning at is a core. It could be applied to any type of tumor. It just so happened we're blessed to have it focus on the sarcoma things, but the single cell technology is applicable to any cancer. So anybody listening and you start to think about, yes, let's get sure, making sure that funding is maintained for all cancer research. And if you only have one cancer research to fund sarcoma. No, whatever you need to do, but fund sarcoma. The other part too is the sarcoma sisters, Wendy Ellen Rose and Dr. Culp, I've asked them, hey, keep posting things because they're fun to listen to and they have interactions and as the other groups as well. I really like that idea too. More people out there getting the word about the importance of sarcoma, you can't underestimate. And SARC is not the only group out there doing sarcoma research. You talked about SFA and research activities and funding, and there are many other great philanthropic groups and patient advocacy groups, which we every bit helps. So let's keep getting the message out. So that's great. As we get close to the end, you've said some things that are important because when you came to MD Anderson, you needed somebody to help guide you, to mentor you during the path. You fortunately came into a good group, sarcoma. You came to a good group at MD Anderson, which is great. And Joe and the team have been helpful for you. Um, the interactions that you have, not only at MD Anderson, but when you and I meet at national meetings or we were just over there in San Diego there, it's more like a family as you go along. But we also want to make sure your career moves along in a good fashion and maybe share how you've used and leveraged your mentors, as you would say, slash advisors, because we got to keep getting more funds for you. So how is all that working in not only within MD Anderson but outside of MD Anderson?
Sure, of course. I think you can't be good at everything, and for me, I'm always thinking like, what's the next thing I gotta learn? Who's the brain I need to pick? And then so I've tried to surround myself with mentors that know more about other areas that I don't know about. And then one of the things that you really need to do when you have these mentors or group of folks that are mentoring you is really have consistent meetings and make sure that there's always a dialogue of things that you're doing, how they can help, how their expertise can contribute to your success in your research. And I think the one thing I found that, to have these meetings is to put it on the calendar. Make sure there's a calendar invite. Everyone accepts it's on there. Otherwise, sometimes, day comes and no one shows up to the Zoom meeting because you don't have that calendar invite. So make sure we have a calendar invite. But the other thing is milestones. You want to make sure that you have milestones every year or every 5 years, even smaller ones for that every quarter that you're hitting. And then if you're not hitting them, what are the feedback or the reasons why the pain points essentially that you need to overcome to try to hit those milestones. And then that and most importantly for milestones is really be accountable. I think. If you say you're going to do something and you're going to do it within a year or 6 months, I think you got to follow up because with mentors, with relationships, with colleagues, collaborations, that's really important. I think when you drop the ball, people notice, and so I think it's really important if you say something, you should do it. If you can't do it, you should be really honest about that. And that's even for mentorship too. Like if you have mentors that you realize over time that you know it's not working out, I think you got to have a conversation and then perhaps find different mentors, or hopefully it does work out in the very end.
I took away, that's excellent. I took away several things. I'm putting up three if those are watching. You said you have multiple mentors, and that's true. Successful people have multiple members. Number two is time is the key. Get it on the calendar. And a component that I think you said nicely that I don't think about all the time is commitment. You say you're going to do something, you're going to do it. Be accountable. And if it's not working, call it out. And it's no stretch in our imagination. We're all busy, but if it's on the plate, I've committed to this, I'm going to do it. I think that's key. That's excellent. I'm going to keep these three things in mind, and anytime I need a mentor, I'm going to keep these three things, get it on the calendar, make sure that there are milestones that are there, and then I'm going to have a commitment as well, which is great. And I have multiple. I look to people, pick their brain, and put it on the table and say, hey, this is what I'm going to deliver. As we end, you said something that's important, 5-year milestone. So, Dr. Truong, when we connect again in our next podcast 5 years from now, what would we be expecting that you have done in the next 5 years?
Yeah, one of the major things that I've really been thinking about, areas of research I want to accomplish, especially for desmoplastic small round cell tumor. There's been a lot of discussion on the genomics, various targeted therapies, but one area of research that I think no one really talks about is the tumor microenvironment. And that might be applied for a lot of various sarcomas, but with DS or CT, there's an aspect of tumor microenvironment, even in the name, the desmoplasia, basically the stroma, reactive stroma with abundance of extracellular matrix. And how that is involved within DSRT is still not clear. We don't know how the fibroblasts within surrounding the tumor cells. We don't know what interactions are occurring, whether they are promoting, the tumor growth. And that's one area of sarcoma science that I really want to uh, go down the line in. And hopefully within 5 years, I'll have some understanding of the role of those fibroblasts. Another area is our models. Perhaps going back to the concept of the tumor microenvironment, most tumor models are usually just the cancer cell lines alone, or they're PDX models, which are somewhat expensive, and some groups don't have access to animal facilities to grow patient-derived xenografts in mice models. And so, one area that I'd like to target is developing these 3D culture models. Some of the things that basically leveraging my expertise from my bioengineering background, my PhD studies. Again, going back to DS or CT, perhaps growing various models where we can culture two or more different cell types like fibroblasts and tumor cells, and understand their interactions that can occur in real time, and tease apart whether that can be targeted or not. Um, and lastly, I think uh, differentiation epigenetics. I think I talked about that earlier about MSCs, how we can leverage that for understanding sarcomas. There's this kind of concept of like cellular reversion. I mean, where you can push cells to different phenotypes. And what I mean by that is like you can perhaps push a very malignant cell to more of a benign state, and how we can really control that. And I believe that's through the epigenetics, basically the DNA and various marks and changes to the DNA. So those are the three major milestones that I want to target in the next 5 years. Hopefully, I'll have some papers, some projects, presentations to be able to present throughout those 5 years regarding those topics.
Great milestones, and it kind of builds when you were talking about it about the 3D. That's what you started here with. You go back to the sugar on the string and getting crystallization, inquisitive. You're starting to have all these scientific questions, and you're going to start to knock them off one by one. And although we didn't spend much time in epigenetics, take a cell and make it behave a little bit better, act more like it's supposed to. So we'll look forward to the next 5 years, and hopefully you will have knocked out all of them or then some. And these are applicable to any type of sarcoma or any other malignancy. So thank you. Dr. Schramm, thanks for your time and sharing what you've done and how you've got here today. Is there anything else you want to share with our audience?
No, I really, appreciate being on here. I'm honored to receive the invite. I know every time I listen, I hear you describe interesting people that you want to hear about, and hopefully, I was an interesting person.
First of all, yes, you are an interesting person, and you're a person that we all should get to know. If anybody has any questions, we'll put it in the show notes how to get in contact with Don Trong here, and hopefully that his name will continue to resonate above and beyond SARC Talk. So thank you.
Thank you.
Well, for those that are listening, we look forward to future SARC Talks in there. Please leave comments that you might have, and please subscribe. And hopefully we'll see you again with our next interesting person in sarcoma. So thanks.
Thank you for listening. Be sure to subscribe so you never miss an episode of SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or for questions, please email us at sarctalk@sarctrials.org. Together, we can find a cure for all forms of sarcoma.